All right, here we go again. All right, so let's get rid of us. All right, so what do we want to do when we want to? How do we make the lungs expand when we're breathing in? So we're going to drop the diaphragm. That's going to increase the volume of the thoracic cavity, and that's going to decrease the pressure of the thoracic cavity or intrapleural pressure. And so then what that will do is as our chest wall pulls out, there is a cohesive force between parietal and visceral pleura. And so the lungs will actually move with the thoracic wall because of the attraction between the parietal and visceral pleura. And so that will help inflate the lung or make the lung volume bigger so that the air can come in. All right, but we have to make sure that we have a minimal amount of pleural fluid, or else then we run into the same problem we run into with a pneumothorax. So let me just show you really quick what I mean. Let me draw that cohesion. So I have fluid. So I have a serous membrane right here. I have a serous membrane right here. So there's going to be fluid all the way across here. And so because there's such a small space between the visceral pleura and the parietal pleura, that the fluid being secreted by both membranes will attract to one another. And so then I'll just have this little film of water going across here. So as this moves out this way, then the lungs will move with it as well. And that will allow the lungs to expand, and then that will lower the volume inside of here, and then the pressure will, increase the volume, the pressure will go down, and then the air will come in. So that's what that means. All right. Okay, so anytime that we have air in the intrapleural space, we're going to get the pneumothorax, and then we're going to get atelectasis because the pressure inside the lungs equals the pressure outside of the lungs. So too much fluid buildup or air in the pleural cavity will cause that. Air can enter through a chest wound or rupture of the visceral pleura. So that means that air is coming in from the respiratory tract, okay, and can happen with pneumonia or vaping, as we know now. So remember, if you want to fix the pneumothorax, then you have to close the hole, and then you got to take the air out. Okay, and so because each lung is in its own pleural cavity, collapse of one lung does not mean collapse of the other one. All right, so ventilation, we know all about ventilation. You can read through this. This is not that important of a concept that I want you to know, but we see resistance present only in the medium-sized bronchi, because as we start to get closer towards the respiratory zone, then diffusion is what's really taking place, and so we end up having very little resistance. Even though they're small tubes, we get less resistance. So this is where asthma is going to be an issue in our medium-sized bronchi. And let's see, what do we want to say? Greatest resistance, medium-sized bronchi. Bronchial diameters are sensitive to neural controls and chemicals. Parasympathetic nervous system and histamine causes constriction, and sympathetic causes dilation. Okay, so we did alveolar surface tension. So this is resistance is going to affect pulmonary ventilation, surface tension. But remember, we've talked all about surfactant. Infant respiratory distress is when surfactant's not made by the babies. And lung compliance, we already know that. That's how easily can they be expanded. A higher compliance makes them easier to expand, but if we have fibrosis or fluid in the way or not enough surfactant or decreased flexibility, that will all reduce lung compliance. Dead space is just simply the fact that of that 150 or that 500 milliliters of air that we breathe in, 150 of it stays in the conducting zone structures. So only 350 milliliters of air actually reaches the alveoli and is actually, this O2 and CO2 is actually exchanged. And then we get alveolar dead space if the alveoli are all too filled up with fluid and we can't exchange. So really dead space is just air that can't be exchanged. Alveolar ventilation is just how fast you breathe, so milliliters per minute, times how much air is actually, milliliters of air are getting out down to the alveoli. The important thing for this is that the depth of breathing enhances alveolar ventilation more than rate, because our dead space is always 150 milliliters. And so if we want to breathe more air, we don't just want to breathe faster, we need to breathe deeper too, or more air is going to remain in the dead space in our conducting zone structures and not make it all the way out to the alveoli. So rapid shallow breathing, so just like panting, is really bad because you're not getting enough air out to the alveoli. But rapid deep breathing, hyperventilation, that's good. All right, so gas exchange, we know all of gas exchange. We did those, and here's that little picture that we've drawn on the board multiple times, and there's all the numbers. And then here is ventilation and perfusion. So PO2 low, blood shunted to areas where it's high areas, arterioles dilate. PCO2 would change the diameter of the bronchioles, and we did all of that. We did that, and there's that picture we did. Here's our respiratory membrane. So during pneumonia, exchange membrane thickens because fluid builds up in the basement membrane between the two epithelial walls. And emphysema occurs when the walls of adjacent alveoli break through and alveoli become larger. And tumors, mucus, and inflammatory material can also impair that membrane. We talked about this briefly. This is blood. A red blood cell will be in the capillaries, in the pulmonary capillaries, for 3/4 of a second as it's passing by the alveoli. And this just shows that oxygen is totally loaded by the time that blood's only been in there 1/4 of a second. So it's very, very efficient loading at 104. Okay, and we know tissues is lower than arterial blood. Transport, so we've done all of that. And here's just, this is in the lungs. So it's that same little activity that we were doing, but it just shows the arrows where they're supposed to go. And then the dissociation curve, we already saw that. So that's that right there. And then here's the difference between resting tissues and heavily exerting tissues. Here's where that 40% more O2 is being released. This just shows the association curve at different temperatures. So here, when it's cold, 10 and 20 degrees Celsius, you can see that hemoglobin really wants to hang on to oxygen for a long time. And that's going to be advantageous to those animals with the mammalian diving reflex that are going to be in cold areas that the hemoglobin carries the oxygen for a long time. But as we get hotter, then it releases it more readily, which is what we want when we have a fever. Okay. Also, this is CO2 levels. So at low CO2 levels, then or high pH, then hemoglobin hangs on to O2, like in our tissue or in our lungs, sorry. And then when we're out in our tissues, we have more CO2 and a lower pH, and then hemoglobin wants to get rid of it. And that's what we want. Okay, impairments of oxygen transport, just different kinds of hypoxias that we have. Anemic hypoxia obviously is not enough red blood cells. Ischemic is the blood circulation is impaired. So if you have congestive heart failure or emboli. Histotoxic hypoxia is poisons that compete for our O2 binding site. Cyanide in particular blocks the O2 binding that we have in the mitochondria and the electron transport chain, and so that's how we die of cyanide poisoning. And then hypoxemic hypoxia is if I don't have enough O2 in the air to begin with. So if I'm at high elevations where there's low PO2, or I have carbon monoxide competing for the hemoglobin, and it's as if there wasn't enough O2 in the air. Carbon dioxide, we got that, and we know all of that, and there's that. And Haldane effect. Don't worry about this graph. Okay, so just this is a good thing to know now for when we get into acid-base balances, the changes in respiratory rate and depth produce changes in pH by altering the amount of carbonic acid that you have in the blood. So slow, shallow breathing is a drop in pH. You're retaining CO2, so therefore you're going to push the graph to the right, or the equation to the right, and it's going to produce more acid. But deep, rapid breathing flushes out CO2, pushes the equation to the left in the lungs with hyperventilation, and you'll have less carbonic acid. Okay, right, we're almost done. So here again, control of center, or control of respiration, where are the centers. So we have the dorsal respiratory group, the ventral respiratory group, and the pontine respiratory group. And so dorsal is what drives it, because see, we're going to the external intercostals and the diaphragm. So that's going to cause inspiration. And this will be, if I want to really forcefully exhale, my ventral respiratory group will be involved, and then my pneumotaxic and apneustic centers will affect either negatively. So pneumotaxic stops you from breathing, apneustic gets you to breathe again. And so we'll just look at that really quick. So here's dorsal respiratory group. There's our rate of breathing. Ventral respiratory group. Apneustic stimulates inspiratory neurons. So however you want to think of this, if you have apnea, you're not breathing, so that would make me breathe, right? I haven't been breathing, so the apneustic center makes me breathe after I haven't been, and pneumotaxic is then inhibiting because maybe I'm breathing too much. I don't know how you want to think of that, to remember that. And then we talked about that, and we've been all of this and the chemo factors, and we saw this, and pulmonary irritants and Herring-Breuer and hypothalamic and voluntary, and we don't need to look at that. And we've been through the chemoreceptors and arterial pH, exercise, high altitude. Okay. And then I didn't know what that video was. Okay, hey, we're done. All right, so that is all for respiration then. And so we will start on Monday with digestive system, and or keep going through our digestive system, finish that off Monday, Wednesday, Friday next week at 9 o'clock. Okay, lab. I'm going to figure that out. So I'll send you an announcement. It's going to be Tuesday because we don't have any other class on Tuesday. So Tuesday, 9 o'clock seems to work good for me. So let's go ahead and say Tuesday at 9 for our 202 lab. So that's everybody Tuesday AM, Tuesday PM, Wednesday PM, Tuesday at 9. But if that doesn't work, then let me know, and maybe we can have 2 lab times. I don't know, but I still have 201 that I have to teach as well. So, okay, other than that, I don't have anything else for you. So, no, because you've had, so Trevor just asked if we start digestion on Monday, should we wait to take that quiz later? No, get the digestion one done right now. I mean, go in and get that finished now that it's loaded in there. Because you guys did that before we left on spring break. Now you're forgetting structures. So go ahead and get that digestive system quiz done ASAP. This digestion that we're starting on Monday is the physiology stuff. So that's for the test a week from Monday. So yeah, make sure you get that digestive system test done right now. Okay, you're welcome, Maddie. And so I hope you guys have a great weekend, and we'll see you at 9 o'clock on Monday morning. And again, if you have any questions, just ask, and you're welcome, Trev. Okay, bye.